So, good morning, everybody. We are going to begin anatomy part two for the MSBS program, and we'll start our journey off for this semester or this 50% of the course with an overview of the cranium, scalp, meninges, and brain. This will be recorded, so after the session ends, I'll post that recording onto the Brightspace in case you need to watch it. Okay. All right. So here are some of the key objectives here. So we tweaked a little bit of the items. This is overlapping with a little bit on the nervous system, but we'll go over just some of the structural organizations of the different parts of the brain. I'll talk about the cerebrum. I'll highlight some of the key areas I want you to know, right? Because there's so much information. It's more important to know some of the key points rather than know all the obscure facts. We'll talk about a little bit on the development, diencephalon, talk about thalamus, the hypothalamus, go over a little bit of the different items in these substructures, and then we'll go over the brainstem. We'll talk a little bit about cerebellum's function, and then at the end, we'll probably get towards the limbic system again. And then the second set of slides, which we'll cover maybe at the second hour, will be on the spinal cord. Okay, so mostly just structural-based images, quite a bit of overlap, and we'll do HoloLens afterwards. All right, so here we go. So the first part of our story begins with just a quick overview of some of the key structures that you're going to see inside the brain. When we talk about the brain, there are these ridges and folds. You've probably seen a model of the brain, seen a picture of the brain. You saw the cadaver images I provided you, but the gyri represents the folds, the sulci represents depressions, and then of course we talk about direction, right? So the anatomical position, model, anterior, posterior. Another synonym that goes with anterior is rostral, towards the nose, and then posterior towards a caudal, towards the tailbone. So as we kind of get into the groove of all the anatomy, I want to remind everybody that's all vocabulary. That's all it is. If there's one secret to understanding anatomy and physio, is if you know most of the vocabulary, you can answer the questions. Chances are when you get stumped, you are not understanding the question because you don't know the vocabulary, right? You're like, is this structure or that's that structure of this? And that's where you have the confusion. So make sure you're very clear on the definitions. Quick snapshot of the human brain. So this is a lateral view. So you can see the exposed textures. You have the lumps. You have these little folds, these creases all over the sulci and the gyri. A few things to highlight, even though this brain model isn't color-coded. Frontal lobe, right, that's on the frontal side, so it matches with the frontal bone. We have the parietal on the back side, temporal towards the lateral aspect of the ears, occipital on the posterior side, and then in the kind of lower head, you have the cerebellum, and then you also have the brainstem, and then connected with it, you have the medulla oblongata extensions to the spinal cord. Right? So we're going to take a look at some of these structures. You recall maybe the most important piece of these brain was the cranial nerves, right? Last half of the class, we went over all the different nerves. It talked about the roots, the function. So we're going to not revisit the optic and the cranial nerves, but we will talk about those structures, okay? So cranial nerves, great. If you remember those mnemonics, awesome. Hopefully you still have a few of those in your notes that you can just pull up. And then another view showing over here. This is the cadaver, right? So right after it, I just wanted to give you the contrast. So a heads up for the practical. Okay, for the practical, I will be using, again, the cadaver images. This one right here is a mirror image of the cartoon, right? So right before that, you have the cartoon. Right after, you literally have the cadaver. So I think this is very nice. You can just kind of compare, contrast it. Obviously not perfect. Some of the gyri, some of the sulci are not nearly as symmetrical, but you still have the same parietal lobe. You still have the frontal lobe, occipital lobe, temporal lobe, and then the lower half cerebellum, brainstem, spinal cord, right? So nice visual for you to look at. This might haunt you just a little bit. Maybe remember this from the inferior view. Yet you can see all the different roots of the cranial nerves, right? Majority of them originate from your pons medulla oblongata. You can see the optic nerve crossing over at the optic chiasm, and then you have the olfactory bulb, so cranial nerve number 1. But this is just a quick refresher of showing you all the roots extensions from the brain. Right after that, I have the cadaver. Okay, so again, for practical, and I'll make a list of the practical images for the exam. But as you're looking through the lectures, anytime you see a cadaver image, those are the ones I'll be using for the exam. Okay, so just that way you don't have to have like 5 different slides. If you like that, I could always tease it out and say these are the 8 images. I'm fine with that. But going through the lecture slide, any one that has the cadaver images, those will be the ones that I will put on the exam. Okay. And then for those that maybe didn't get the memo, I'll be making a list of structures. So we're going to have an anatomy list. Every class that we go through, we're going to post like 10, 12 structures. And those are the ones you got to look for. And that's it. Okay. So for example, I'll just list olfactory bulb, frontal lobe, temporal lobe, occipital lobe, medulla oblongata. And then you got to be able to identify those ones on the image. Fair game? Sounds good. So it's just point and look. Okay, so here we go. So this is another view. So we'll start off with a little bit on the brain development. Right? So this is a whole topic in itself. But by the late to 4th week of embryonic development, there are these structures called the vesicles that begin to develop. And what's interesting is there are only certain parts of the brain that will develop into secondary structures. The midbrain, or the brain stem, that stays as the same structure early on till even adulthood. Dividing the brain up, we have 3 kind of divisions: the forebrain, the frontal region, the midbrain, and the hindbrain. The terms that we use to describe them are the proencephalon for the forebrain, mesencephalon, midbrain, rhomocephalon for the hindbrain. Right? So just remember the mesencephalon with the M, midbrain is M, pro goes with the 4, and then rhomocephalon is the back. Okay, so think of it, R as the later letter in the alphabet, so it's towards the posterior side or towards the back end of the brain. Moving on, then by the 5th week of the embryonic development, there are these key secondary structures that develop. Okay, so this is, again, not the coolest topic of them all, but the embryonic development does give you a hint at how the brain eventually becomes its final form. Telencephalon is going to be the origination from the proencephalon, and the key structure is it forms the cerebrum. All right, so as I'm going over this, there's a lot of terms. What you need to do is word association. Okay, so when you're doing the MCAT, when you're in medical school, when you're trying to take your boards, I will probably say half of the skill set is association. You're going to read so much stuff, and you're going to see so many PowerPoints that there's no way you're going to memorize everything. But you need to remember what goes with what. Right? So this receptor, this hormone. This structure, this part of the system. Right? You got to associate what with what. Telencephalon goes with the cerebrum. And then the diencephalon is going to be the creation for the thalamus, the hypothalamus, and the epithalamus. Okay, so I'm just going to go ahead and key them together. Right, so as you're forming your notes or using the slide deck to create a study guide or highlights, make sure you know what goes with what. Out of all 5 of these substructures in week 5, the only one that does not have a secondary new structure is the mesencephalon. That literally becomes the midbrain. The midbrain stays as it is, even on the early stages. So whatever you start off with, you create that final form. Okay, so I'll put a little color here to it, but the mesencephalon is the only one that does not have the ability to create a secondary final form. Metencephalon is going to be the rhomocephalon that forms the pons, and then the cerebrum, parts of the cerebrum, and then the last one, the myelencephalon, forms the medulla oblongata. Okay. So just be mindful of which final form. So the ones that I circled here are the structures of the final form of the brain, and then the previous structure are going to be how it originates. Okay, so as you're going through embryology, and this is just a snapshot, there is a embryology course in the medical school where you're going to hear a lot about these different subcells and structures that originate and become the final form, right? And a lot of changes occur in that week 4 to 5. So that's one of the key windows. During fetal development, 4 to 5, 5 to 6, there's a lot of changes that occur that are also noticeable. Okay? So this is just a cartoon image showing you the development of your neurotube. So early on, you can see that purple color region is the proencephalon. The mesencephalon is that yellow region, the midbrain, and the rhomocephalon is what? Is that hindbrain, okay, the posterior side towards the cerebrum and cerebellum. And then down you have the spinal cord. At this phase, when you look at any mammals, any species, they're all pretty much the same. You can't really detect in terms of just the neurotube, but eventually these different origination structures are going to give rise to the secondary structures, and that can give you a hint as to what kind of a brain size and brain development is going to have. Okay? So that's just your embryology of the brain. Eventually, we become a fetus at week 13. That's going to be the timeline. So what I went over today is you should be mindful. Week 4 to 5, there's big changes. And then from week 5 to 6 to 13, there's a big change. Okay, so every week there's changes, but we're not going to study every single week. But just know 4 to 5, 5 to 13, those are some big windows of maturation and changes to your brain structure. This is a nice chart that kind of overviews the entirety of the process from neurotube all the way to your primary vesicles of the brain and secondary. So I like to use these to kind of help you review the key items when you're studying. Over here, you can see the key terms as well. So telencephalon, diencephalon, mesencephalon, metencephalon, myelencephalon, and then on the right side, the structures that it forms. Okay, so this is the same exact stuff I just showed you in the previous 4 slides. Kind of like a study guide, cheat sheet for you when you're reviewing. Okay, so when you're studying these set of slides, and I made an intention to kind of give you the high yield. That's what they always want, high yield. It's whenever we have sessions at the medical school, there's always a lot of information, right? And the job of us facilitating or instructing your class, running your class, is to take that content, and this is how I see it, distill it down, give you the stuff that's the most important so you don't have to go digging around. Right? Because you can spend hours studying just by digging around content. And you realize you're like, oh, so which one should I go with? So I'll try to give you in the format charts that kind of focus you onto what's most important. So just make sure you know which ones correspond to which ones. Okay? All right, so telencephalon is to cerebrum. Diencephalon is to the thalamus, epithalamus, hypothalamus. The midbrain basically doesn't change too much, but the different colliculi. These are so the superior inferior colliculi, by the way, guys, these two structures are integral nuclei inside the midbrain that are used for visual and auditory reflexes. So when you see something and hear something, the signals that go through to the brain, there are certain circuits that are hardwired. Those are two hardwired for visual and auditory. And then the metencephalon becomes the pons and cerebellum, and then the myelencephalon becomes the medulla oblongata. Okay, all right. So that's embryology in a nutshell. That's all we're going to go over. So hopefully that gives you a dose of the development of the brain, early stages. Now the second kind of set of content, it's just going to be a little bit of overlap from our previous session. So we're going to start off with the fact that the brain, your spinal cord, is composed of 2 kinds of neuron sets. You have the gray matter, white matter. And gray matter means what? Which ones are myelinated? The white one, right? So myelinated means what? It means it has the lipid coating around the axon. Does the myelination provide a quicker conduction or slower conduction to the neuron's signal? Quicker, okay? So some things to recall is that myelination indicates that it's a faster signal. There's a saltator conduction where it skips from one node to one node. Those are usually also the bigger diameter ones. So the bigger the axon diameter, the faster it conducts. A few things to be mindful of is that they house the motor neurons. They also contain the interneurons, cell bodies, right? Those dendrites. Those are all the substructures of a single neuron. Most of the gray matter creates what we call the cerebral cortex. Okay, that's the surface of the adult brain, where all these kind of lumpy, dented structures stay within. And then within the deeper parts of the brain, we're going to have the white matter. And then inside, you're going to have clusters called the cerebral nuclei. These are dedicated neuron sets that have these neural bodies. The white matter, which we said is the myelinated axons, and they're deeper to the gray matter. Just remember, in the spinal cord, it flips around. So in the brain, gray out, white in. In the spinal cord, white out, gray in. Okay, so there's a transition there. Most of the crossing over of these neurons occur right at the midbrain. Okay, so as you come down the midbrain to the spinal cord, there's a crossing over. So your left hemisphere corresponds to your right body. Your right hemisphere goes to your left body. Okay, so there's a lot of interesting phenomena that occur as you transition. This is a nice view, and I'll zoom in a little bit. So you should be able to, can't move this one. Okay, so when you get a chance to look at it on your screen, you might have to get closer to it, but you can see the top picture is a kind of a corona cross section of it. You can see the outer gray matter, inner white matter. Notice that there's also going to be these dedicated kind of bundles. These are the basal nuclei right here. We went over these last course, but we'll go over them again. the putamen, the claustrum, amygdaloid body, but these are dedicated bundles of the nuclei that have a function. But just for now, cortex is on the outside, the nuclei are deeper within, and then the inner white matter. Over here, right below it, this view, I'll zoom into it in a second, but you can see the cross-section of the cerebellum. Again, also, the outer layer is darker, is the gray matter, and then the inner is the white matter. Notice over here on the spinal cord at the bottom, so now compare those two images on top with the one that I want to circle with the red. You can see it's going to be what? The white matter is on the outside, and the gray matter is on the inside. Okay, so there's a flop in terms of the gray white matter. All right, so key structures of the brain. We'll start off with a little bit on the cranium. Key functions that maintains protection for the brain structures. It's cushion, right? It's a soft structure. You don't want it to be pounding against the hard surface of the cranium. And then, of course, we have the CSF that maintains buoyancy, the cerebrospinal fluid. Fluids around generated by the four ventricles, the first, second, third, and fourth. And then, of course, majority of your nervous system and your brain, CNS, is layered and walled up by the blood-brain barrier. Right? These are epidermal cells. Remember this? Epidermal cells. We have the astrocytes, cells that form the boundary, right, of your nervous system. Okay, so let's take a look. So highlighting some of the key structures here. We went over this also briefly, but the cranial meninges, these are soft connective tissue that serve as the boundary and protective walls of the brain. They separate and tease apart the brain from the cranium. So you're not just going to have this solid, brain and then the soft tissue of the compounding together. And what's important also is that there's also a lot of blood vessels that circulate, also CSF. Within the meninges, there's 3 layers. Okay, the first layer, the deepest one, thin layer called the pia mater. This is what touches the brain itself. The spider layer, arachnoid mater, which you remember also matches really well because it has this web of fibers. Right? So arachnoid sounds like arachnid, spider, web of fibers, and then the fibers are called the arachnoid trabeculae. All right, so a lot of the terms give you a hint, right? You're not in the dark. Just remember, arachnoid mater creates the arachnoid trabeculae, which is basically like a spider web. Okay, spider web in the middle layer. The dura mater is the outer tough layer. Highlighting here is the outer layer is subdivided. Okay, there's 2 subdivisions. There's the superficial parasotial layer. That's the one on the outer side. And deeper within, you have the meningeal layer. Okay, so really, if you think about it, it's like there's 5 layers, okay, or 5 terms so going from the outside. But there's 3 main layers. The outer layer has 2 layers. Here's a picture. So if you zoom into an image, you'll see this is a nice little cartoon. So kind of zoom into one cross section of the cranium. You can see the skin of the scalp. Right below it, you'll have a little thin layer of the parasodium. Parasodium is the outer film of any bone. Okay, that's your outer layer of the bone. Then within that layer, or underneath that layer, you have the bone itself, so the cranium, right? So whatever, frontal bone, temporal bone. And then right below that, you're going to have the dura mater, which we just talked about, right? 2 subdivisions is what? What's the outer layer called of the dura mater? Parasitial, okay, so P, and then meningeal is going to be the inner layer, right? So just always have that in mind. One way you could kind of, I guess, get comfortable with the layering is think about which letter comes later in the alphabet. So P comes after M, so it's going to be higher. So it's the outer layer. Meningeal will be the deeper layer. I don't have any cool mnemonics for this one, unfortunately. All right, so there are there are quite a few for the endocrine, so don't worry. I will have some mnemonics to help you kind of key things together. But for the brain, it's a little bit, you got to raw it. And then right below that, you're going to have the arachnoid mater, right, the spider layer. And then right below that, we have the pia mater. Notice that between each layer, there's also a small potential space. Okay, so between the dura mater and the arachnoid mater, we have what? This layer is called the subdural space. It's a small gap, a little bit of cushion. Between the arachnoid mater and pia mater, we have the subarachnoid space. Okay, so if you get really picky, or if you really want to detail it, every space also represents a layer, right? It's a gap. It's a space between the two. Okay, so just again, running through, we have the dura mater, subdural space, arachnoid mater, subarachnoid space, and then pia mater. And then you have the actual brain. So your brain isn't just smashing onto the skull, all right, when you're shaking your head. There's a cushion, there's a web, there's a little bit of space, and allows for forgive. So within the meningeal layer that we just talked about, it extends into 4 different divisions of your cranial cavity. I had this on one of the previous slides last in the nervous system towards the end. You may recall this or you may not, but it's all good. 3 key structures that I want to highlight that create these depressions and grooves that allow the brain to sit on. The first one is a projection that goes from the longitudinal aspect. So that means from front to back, separates the left and right hemispheres. But that's called the falx cerebri. Okay, so these are films, like webs or partitions between different divisions of the brain. Then we have the falx cerebelli, right? Cerebelli, don't get confused with the word, cerebri and cerebelli. This one separates only the hemispheres, okay? This one goes longitudinally. And the last one, which you have to know, okay? And I'm just giving you a little wink and a little hint here. It's gonna be on the exams, gonna be on the quizzes, is your diaphragm sali. This is the saddle, the film, where the pituitary gland and your hypothalamus sits, right? So this is literally the center of your brain. If you had to find the core of your brain and you pin it out, that's what it is, okay? So diaphragm sali, that's a saddle, basically the saddle where the gland sits on top of, okay? And anybody remember which bone this is on? We went over the bones, so that's why I expect you guys to have at least some, you heard before, it's in there. You just gotta go and retrieve it, right? It's like you're trying to find an old set of notes in a cabinet or cleaning out a closet kind of thing. Which bone? Frontal, ethmoid, temporal, sphenoid. Sphenoid. There we go. Okay, so I know it's early Saturday, but remember the bat bone? Remember the Batman has a wings, greater wing, lesser wing. Right in the middle, you have the sella turcica, diaphragm sali, little depression. That's where it is. Okay, so that's the little septum region where it coats over in the spinach. It's a partitions of brain. So it's key structures for support. Now, within the brain, you also are going to have a duro partition. So the duro partitioning are also known as the durovenous sinuses. These are basically layers that also cushion. 3 of these are very noticeable. The first one is called the superior sagittal sinus. Okay, so use the terms, give you an idea. Superior means towards the top. Sagittal is going to be dividing in the mid-plane, so left and right side being split. This is going to be located towards the superior aspect of your falx cerebri. I'll have a picture, so that way you can see where they're pointing at. Then we also have the inferior sagittal sinus. That's going to be towards the inferior margin of the falx cerebri. The last one is the occipital sinus. Right? That gives you already direction. So that's the posterior border of the falx cerebelli. Okay, so with all the terms, you're like, you got to see this. A lot of this stuff, especially anatomy, is visual. Right? Once you see it, you can kind of have an idea of this is that partitioning, this is the region that we're trying to talk about. Over here, this is a very clear image. I like this one. Zoomed it in. Try not to put too much content. So notice in this set of slides, I try not to put too much content with an image. So that way, when you're looking at it, you just focus on the image. Okay, so I don't know if this is going to help. Previously, I always will. When I do the medical school, I always have an image and I have like the content right there. So you're reading it as you're looking at it. That makes sometimes the image a little smaller. But for anatomy, it's a lot of visual, right? You have to see it to know where it's at. So the first thing I'll highlight, falx cerebri. It's a big film. You see the big film, the kind of partitioning film from the anterior all the way to the back. Okay, it's like a web, okay? Partitions. The other one right below that is the falx cerebelli. This one you can see kind of spans the posterior aspect. Actually, let me use another color so you can see it. Okay, so it's the entirety of the posterior region, right? So again, this is not 3D. This is a 2D image. But you want to imagine this is a film, a partitioning, a cushioning for the brain. And then remember this guy, diaphragm sali, that's the sella turcica, literally on the sphenoid bone right in the middle. Right there, okay? It's a little partitioning right in the middle. Okay, so kind of three color codes for you. There's an orange, there's a green, there's a red. These are the key ones. And then there's one that I did not put into the slide deck, but in the middle, right kind of behind the midbrain, we have the tentorium cerebelli. That's another film. So these are the key films that cushion your brain. Right? So when you're sitting the brain into the skull, into the cranial cavity, it's not, again, sitting on a hard surface. It's being cushioned. Okay, it's being anchored. It's being supported. All right. This view on the left-hand side is the cadaver view. So again, another little hint here. So as you're looking at it, maybe put a star. This could be on the practical. This is a cadaver view of that same image. Obviously, it's not perfect, right? Because you can see some of the asymmetrical, some of the film is already kind of faded off. The dye for the veins aren't perfect. But some things you can see is the falx cerebri. Okay, I'll circle that one. Make sure you can identify that that's a big film. Diaphragm sali, falx cerebelli. Okay, fair? So again, I'm not going to try to overload an image with 10 or 20 items. The key ones, I'll generally give you like a little circle or give you a little nudge. Those are the ones you want to look at, which means for this week's anatomy list, these three will definitely be on there. Okay, so when I make that list and I post it on Brightspace, falx cerebri, diaphragm sali, and falx cerebelli, I'm telling you right now, those will be on there. Okay, so as you're going to start off 2024 strong, I would recommend today is first week or first day of anatomy. Get your notes, get a folder, get your PowerPoints, get your PDF or your iPad, start creating your notes, right? Keep going. Okay, so we had a good break. Winter break is over, back on the grind. Okay, all right, cool. Next one, what we have here is now we go over the ventricles, right? So another structure in the brain that's very critical is the development of the CSF, cerebrospinal fluid. This fluid reduces the buoyancy by, I believe, like 45-50% of the brain's weight. So it gives you the ability to not only bathe the brain, but it's generated through the ventricles. There are 4 of them. The first 2 are what we call the lateral ventricles, 2. Okay, so 1 and 2. These ones are going to be housed in each of the hemispheres, the left and right side. There is, again, another partition, a little divider between the left and right side of the brain. Okay, for that creates these ventricles. That's called a septum pellucidum. The third ventricle is right in the middle, and as it makes sense, because if it's in the middle, it's going to come from the diencephalon. Okay, that's the middle region. It then has a little opening that allows a passage of the CSF to drain through, right? Because if it's housed in the upper chamber, it has to be able to travel. To go from 1 and 2 to 3 and going downwards, we have the interventricular foramen. This is a passageway for that fluid. All right. And then going from the 3rd to 4th, the connection is called the cerebral aqueduct. All right. So very simplistic. I pinned out these key items for the drainage of passage of the fluid. And then the last one will be the 4th ventricle at the very bottom, but this basically merges down with your spinal cord as a connection.
So with the 2 lateral, you saying this
so when you label them 1 and 2, there could be genetic variations. So some people may have the left hemisphere draining more. So you might call that the 1, and then 2 would be the minor one. But the best way for that's why I put it here. I didn't write 1 and 2. I just say lateral. But just remember, literally, if you've seen there's this pretty cool model. I think we might even have it, but it's basically like a Star Wars TIE fighter. Have you seen Star Wars before? The TIE fighter, it literally looks like a TIE fighter. The 2 ventricles are like the 2 wings on top, and then the middle part is where that little capsule sits. So if you've seen a Star Wars TIE fighter before, okay, it's like that big airplane with the little ball in the middle. The lateral ventricles are the top wings. 1 and 2 are tricky because if you point out, it's like you don't know which one's dominant. So I would always refer them to as lateral, but that's technically the 1 and 2. The third one is right in the middle, and then the 4th one's on the bottom. I'll have the image right now to show you. So right here, you'll see kind of the imposed image in this visual. It's not perfect, but you can kind of see the TIE fighter kind of vibe, right? So you have the 3rd ventricle right here, 1 and 2, or the lateral. I prefer the lateral term. Some of the older books will say 1 and 2, but it's very misleading because you don't know which is the left one 1 or is the right one 1, is the left one 2. So lateral, 3rd ventricle, cerebral aqueduct, that's a drainage site. The interventricular foramen, you can't really see that really well in this picture, but it's right here in the middle. There's a little opening where the lateral can drain to the 3rd. Cerebral aqueduct, that's going to be drainage for 3 to 4. Okay, so make sure you know the name of the connection. This is a circulating fluid that gets generated generally at the night when you're sleeping, and there's a lot of studies that are trying to analyze when your CSF gets regenerated and recycled, but that's the detoxification of the brain. That's why sleeping is so important, is that when you're sleeping, your brain actually will cleanse and detox. Waste management goes through your liver, but that's the drainage for the brain. The lateral view is kind of cool, but you can see right here it has a big wing. So this is the lateral one. So this could be the 1 or 2. The third one is right below it. Okay. And then the fourth one's right underneath it. And then that little passageway is the interventricular foramen, and that little tube is the cerebral aqueduct. All right. So if you were to take this structure out of the brain by itself, it looks like a little jet plane or Star Wars TIE fighter. All right, cool. So key functions of CSF. Okay, so as we're going over this, what does it do? Cerebrospinal fluid plays a role to circulate all those ventricles. It is housed within the subarachnoid space. Go for it. It's the drainage, so it's an opening. Yeah. So it's a foramen. That's why it's called a foramen. It's still a little hole or a little opening. But if there were issues with the CSF, oftentimes there could be maybe like a structural genetic mutation where you have a deficiency in one drainage fluids reduced. But the key functions here is that it maintains the health of your CNS, right? So number one, buoyancy helps you reduce the weight of the brain, so it helps you keep it afloat. Protection, cushion fluid. It also provides environmental stability, right? So the ion, maintaining the sodium-potassium ion ratio. It also maintains some of the waste management. And then the structure that generates or nourishes the fluid is called the choroid plexus. This is housed within the ventricles. So the cell type inside the ventricles is called the choroid plexus. This is a layer of epidermal cells that form the outer wall, but this is a dedicated cell type. And this is the histology or the machinery that will create the fluid for the brain. All right. As a result of this, then of course we have that outer coating that protects your brain from other invasions and regulations of viruses and bacteria, but BBB, the blood-brain barrier, this is what regulates what gets to go in and out. Around the endothelial cells that create BBB, we have astrocytes. Right? And just recapping real quick, astrocytes are these kind of star-like projections. They anchor blood vessels together. They have these paravascular feet, which can attach to nearby vessels to recruit growth. Right? So angiogenesis, neurogenesis, really, really important for all the astrocytes to be healthy. Most of the time when you have cancers, right, of the brain or tumors, it's the astrocytes. The majority of the time when you have brain tumors is because of these cells being too active. There are kind of some areas of the brain where there are minor unique features, okay? And this is where there are reduced or almost no BBB at all, no blood-brain barrier cushioning. The first one is the choroid plexus. The region that makes the CSF, there really isn't a layer, all right? Number two, which is also very important for endocrine in another 2 weeks or so, is your hypothalamus and pineal gland. These two structures are the only ones that don't have a dedicated boundary. Makes sense, because the hypothalamus is your tier one of all the hormone signals. They release a chemical, right? For example, TRH, thyroid releasing hormone, floods down to your pituitary. Your pituitary can then make TSH, goes down to the blood vessel, go to the thyroid gland, and then you make TPT4. So there's this communication aspect that needs to be quick, no blockage, right? The hormone goes out into the bloodstream, gets to your body, so it goes down through a command system. Right? So just make sure you're knowing that. Hypothalamus, pineal gland do not have BBBs. Cartoon image showing you the astrocytes in case you don't remember. It's been a while. You have the astrocytes shown here in green, the nucleus, but they have these paravascular feet which wrap around the nearby vessels, anchor. They are thought to be critical for neurogenesis. A lot of the studies on brain development. So the biggest talk was like, oh, your brain doesn't regenerate anymore. And some places of the brain, hippocampus, do have neurogenesis activity. When you're using it, you can still get some neural link together. The key point is that the astrocytes need to feed that growth. So when astrocytes are not able to adhere the vessels close by, then you're going to have regression and no blood support and also no development. All right, so the next couple of slides that we're going to go over are going to be in each of the structures. Okay, so each of the parts of the brain, I'll talk about some of the key items to look for. Be mindful of the key functions. So the first one is your cerebrum. Okay, this is where all the higher mental function capacity occurs. You have the thought, conscious, intellectual ability. The region contains the outer cortex, inner white matter, and it makes up all this gyrosphere. Okay, so this is what you see when you see the brain. Right? When you think of the brain, you're thinking cerebrum. We have the two hemispheres, left and right side. In the middle, we have the corpus callosum, which links them together, right? The left and right hemispheres. And I believe I even hinted that various populations and certain kinds of, if you're right-handed dominant, left-handed dominant, if you're ambidextrous, if you train your brain a certain way, the corpus callosum can develop in a way where it links the brain to better suit for multitasking. Okay, so the corpus callosum is believed to be variable. Right? Not everybody has the exact same connection between the two hemispheres. Here is a snapshot. This one's nice, a color-coded one. Right. Last time we talked about nervous system, I did point out that there is one lobe that's not visible. Which one is it? Insula, right? And then what function does it take, or what kind of stimuli does it receive and process? Anybody? Huh? Taste, right? Smelling really good. So taste. Okay, so we're right here. The hint is on the screen. The gustatory complex. So that's your taste. Insula is the only one that's non-visible to the cerebrum. You have to tease it open right between the temporal lobes, and then you'll see it. But this region is what processes the taste stimuli. So write that down, the gustatory or taste stimuli. How about the temporal lobe? What does this lobe respond to? What stimuli does it pick up? Hearing, right? Auditory. Okay, so auditory processing. Okay, make sure you're aware of that. You're going to see questions on the quiz or exam. I'll just ask, hey, which of the following lobes processes this region, right? So let's say a patient comes in, for some reason they can hear the ossicles and the tympanic membrane is all perfectly fine, but somehow they can't process the hearing information. Which lobe is most likely maybe impaired, right? And then you just know which one, okay? Occipital, this one should be straightforward. Visual cue, visual cortex. You also have association areas as well. Parietal is going to be what? The somatosensory. Okay, so all of the muscle, sensation, temperature, soft touch, light touch, itching on the skin of the body, that's going to be parietal. And frontal lobe, you're going to have two key items here. We have the Broca for motor speech, so the processing of speech, as well as parts of the visual field of the eye, and also the primary motor, the muscle control of responses. Okay. So these images and these pictures, the reason I don't put text on there now is I want you to focus on the image. Look at what's telling you, right? That's really the value. Whenever you see a picture, it's more organized, it's streamlined, it's there for you to condense information in a digestible manner. Okay, so highlighting these three terms, and you may have seen this a few times, but make sure you're aware that motor area or motor function just means these are voluntary controlled aspects of the brain utilizing muscle. Okay, motor. Sensory means they pick up senses. Okay, that's going to be light touch, pressure, stretching, but any kind of sensation. Association areas are going to be the ones that can integrate both or store information together. Okay, so when I say, or when you see association areas, I don't want you to think, oh, it's like whatever that means. It means it's able to integrate both motor and sensory information at the same time. Okay, where there's a little more multifunction in those aspects. So the primary motor cortex that we said, this is going to be for voluntary control of all the skeletal muscle within the precentral gyrus, that's location. Axons go all the way down to the brainstem and your spinal cord, so the whole body. Okay, so one connection. And then you may have also seen that homunculus where we briefly talked about the lower parts of the leg corresponds to the upper part of the brain. The lateral part of the cerebrum goes to the upper part of the face. So it's not proportionate. You have a lot more parts of your primary motor cortex dedicated to your lips, to your armpits, to your genital regions, than to the back of your feet. Right? It's disproportionate. Okay? So your body isn't evenly distributed. It's not symmetric at all. Cool. We're almost at break time, okay? Don't worry, almost at break time. So this is that picture that I was referring to, the homunculus. So you can see just real quickly the primary motor cortex. If you were to break up and dissect open this cerebrum and look at each of these subdivisions of the sulci gyri, the toes and the knee and the leg correspond to the upper part of your brain. As you go from the top to the lateral side, you're going down, basically from the bottom of the body, the lower limb, upwards, right? So you're traveling upwards as you're going from the top down. Then you have the shoulder and arms right here. Notice that trunk, shoulder, arms. Then we get down to the lower limb, the upper limb, sorry, the forearm, the fingers and the hands. Notice over here, so on the lateral side of your cerebrum, look at this. Look at the disproportionate. The neck, the eye, the face, the lip, the tongue, the pharynx. Next week we're going to talk about the pharynx and then the throat region. But what you'll see is that the region I circled in blue pretty much is a lot more than any aspect of the trunk or limb by itself. Right? You can see that. So your brain dedicates a lot of precision and kind of sensory input understanding to those areas. Same thing for responses, right? So for the motor areas, there's two regions I'm going to highlight. The motor speech recall, also known as Broca. This one works close with another part in the temporal lobe called the Wernicke's, right? That's for sensing of hearing and sound detection. Those two communicate quite frequently, but this is important for creation of vocalization, controlling your larynx, moving your epiglottis to be able to phonate. And then we also have the frontal eye fields inside the frontal lobe. Okay, so this is for regulation of the eye movement as well as being able to understand what kind of visual cues are coming your way. And that then sends signals to the brain for routing. All right, so let's take a quick break. Looks like everybody's kind of drowsing off. Pause for a second, get some air. We'll freeze for a few minutes, stretch. To be mindful of is we have auditory association, which means that this is the areas of the temporal lobe that process auditory cues, auditory reflexes. Generally, this is also connected to the visual association areas. The two subnuclei that I pointed out in the beginning of today's talk is called the superior inferior colliculi. Now, these are hardwired neural sets that will send signals through auditory or visual cues. The other part that matches with speech and understanding of sound is the Wernicke. So Broca, Wernicke. Motor, hearing. Superior colliculi, inferior colliculi. You have the visual auditory reflex. Okay, these are some of the real important pieces of the brain that are dedicated for understanding sound and then being able to phonate. Typically what happens is in Wernicke's region, it's usually located in the left hemisphere and it overlaps with the parietal temporal lobe. Interesting fact here is that different genetic variations will have slightly different positioning of the Wernicke. And that could change the ability for processing of sound to speech. So if you can't hear something, chances are you're not able to phonate. There's a connection that happens between those two. And then next, what we have is the nuclei. So this might be a recap of some of the key nuclei in the basal ganglia. 4 structures I want to highlight. Make sure you're mindful of what they do. I try to make it really simplistic. You could expand on this easily. The caudate nucleus key function is for coordination of gait and movement. Amygdaloid body is for emotional, mostly fear. So fear is a raw state of emotion that is controlled by the amygdaloid body. The lentiform, this is where you have 2 subdivisions. The putamen, there's actually internal, external, but we'll just go putamen. And also globus pallidus. This is for muscle tone, muscle movement. And the claustrum is for composing of those different stimuli together. Okay? I'm not going to expand on each one, but just have a brief understanding of what each of these nuclei do. This whole set that you're seeing right here on the screen is called the basal ganglia, also known as basal nuclei. All right, so basal ganglia, basal nuclei. I know I went over this because we had a cross-section of an image, and I showed you the different lobes and the internal structures. Here is a visual for the picture. This is not a cadaver image, so this is just kind of FYI. You have the lentiform, which includes your putamen, globus pallidus, caudate nucleus, amygdaloid, and the claustrum. Right? So they're deep within. They're bundled within the kind of darker stained regions of these neurons. Here is the cadaver image. So for everybody, I'll put a little star to it. This is the one that will likely show up on the practical. Make sure you're mindful of these structures. So again, you might see this on the list. I'll try to give you an estimate about usually 12 to 15. Okay, so I don't think we decided on a number, but for my list, I'll generally have somewhere between 12 to 15 per topic. So that means by the first quiz or the first exam, you'll have like maybe 40 to 50, right? So 12 to 15 per, at least about 40 to 50 items to look at for the practicals, right? So the images. So I really hope, because when I was looking at some of this analytics for the final, I noticed there was a couple, maybe some images were a little bit vague, but those practical images, those are the ones I hope it's like the easy ones. Right? So the easy, when you take an exam, you get those kind of lower lower yield kind of the easy fruit kind of questions. Like, oh, I see this one, I know it. I want the practical questions to be the easy ones because it's just picture and a name, right? A picture and a name. Okay, so caudate nucleus, the putamen, globus pallidus, and then the claustrum amygdaloid body. All right, so 5 items for the cerebronuclei. Now let's quickly go into diencephalon. The 3 parts are the epithalamus, thalamus, hypothalamus. And I'll quickly go over what each of these structures are. So I'm going to skip this image. We'll have plenty of HoloLens in a second. Starting with the epithalamus, the key functions here is this is what forms the upper border of your diencephalon. The key structures include the pineal gland. Okay, this is one of the endocrine glands, which we'll talk about. Main function is to secrete melatonin and regulate your sleep cycle circadian rhythm. Right? So this is exactly why when you don't see a lot of sunlight and a lot of UV, you tend to feel a little bit more down, a little bit slower, right? This is Seattle for everybody. So when I first came to the United States, my impression of the United States is raining every day. Seattle, right? Gray, you don't see sun. And then there's also a subdivision within that hypothalamus called the habenular nuclei. The function of the habenular nuclei is this is the connection between the central part of diencephalon to the limbic system. Okay, so this is a connection. As we're going through this, I'm going to highlight the key nuclei that relate all the brains together. Your brain is integrated. Right? Even though we're going over these in isolation, this is the diencephalon, this is the rhombocephalon, this is the cerebrum. They're all interconnected. Okay? A signal can pass through, and it can end up being processed in one lobe. The thalamus, super important. You should remember that this is the intersection for everything in the brain. The masses of the routing are called the interthalamic adhesion. So the word is thalamic is in there. That should give you the clue. If you see this on the exam question, I say, which of the following is a structure that allows the thalamus to route signals out? Interthalamic adhesion. Okay, and then you'll see different masses depending on the picture, but sometimes we refer to them as thalamic nuclei. Right, here's a picture, literally right in the core of the brain. This big circular kind of oval shaped structure. If you tease it out, different, and this is color coded, but different aspects of that thalamic nuclei have different dedicated groups. You don't need to know all these subgroups, but just to kind of give you a snapshot, we have the anterior, lateral, and posterior. Okay, so there's 3 divisions, and then within each division, you're going to have a dedicated kind of route to a different part of the lobe for processing. All right, everything except for smell passes through the structure. Okay? We went over this as well. So the only one that gets to bypass the thalamic nuclei is your sense of smell. It goes to the limbic system as a straight shot. Of course, the one that is the king of endocrine and the top tier 1 is your hypothalamus. Make sure you know this. Hypothalamus is the key structure that plays a role for not only communicating to the pituitary gland, but it has a stalk-like junction called the infobitula. Okay, that's the juncture between the two. It's literally like a little stalk. Some of the general functions and some of the nuclei include all of the ANS control of the body, all of the master endocrine hormones that we talked about. So anything that has a releasing hormone is made here. Okay, usually this is the first line of command. Body temperature, food intake, sleep cycle, all that stuff. So that's why it's everything in both. So the master command system of the body. Then we move down to the brainstem. Brainstem is connection of the forebrain to the cerebellum, and then eventually also to the spinal cord. All right, so we're working our way from the cerebrum to the middle, to the brainstem, to the spinal cord. Contains all the reflex centers. We also have 3 subdivisions: the midbrain, the pons, and of course the medulla oblongata. So some of these respiratory centers, apneusic for regulation of breath, cardiac centers, pressure centers, and so forth. Over here is an image of the brainstem by itself. You can see a couple things. Number 1 on top, you have the thalamus, the oval thalamus. Optic tract. Going down, you have the peduncles. These are the juncture between the thalamus down to the brain. And then we also have the olivary nucleus that's not shown here, but the bulges that you see. So if you look closely at the stem, you'll see these kind of ridges and bulges. These bulges house the nuclei. Okay? There's actually over 100+ different nuclei. There's some that are still being analyzed. There's one that's actually really fascinating called the red nuclei. That's for alertness and also some of the almost when you wake up in the morning and your brain boots up, the adrenaline center of the midbrain is right there. Over here, let's take a look. And then over here we have the midbrain. So some of the things that I point out, so aqueduct, already went over this, is the drainage for that fourth ventricle. And then below that we have the juncture to the primary motor cortex and spinal cord called the cerebellum. Cerebellum is the juncture for the motor control between your spinal cord and then your upper parts of the midbrain. Okay, so it's the link. And this is also where crossover occurs. Around this region is where all those motor nerves get to cross over. And so they call it decussation, where you cross from the left to the right. Okay? The crossing over occurs in this midbrain section. Well, in the midbrain, there's also going to be the dopaminergic center. There's a lot of substructures that we're just going to quickly highlight. This region, called the tegmentum, is the powerhouse of all the dopaminergic pathways, and the specific nuclei, called the substantia nigra, is what generates the majority of the dopamine in the brain. Okay? A lot of the studies have pointed out that emotional control, pleasure, reward system is all right here. So when you feel good, you're addicted, when you feel engaged with anything and you're happy about it, this is the region that's constantly flooding dopamine into your brain. You have the D1, D2 receptors. They bind. Likewise, the degeneration of this region is also the culprit leading to issues of Parkinson's as well. Okay, so there's variations of how severe it is, but this is the powerhouse of dopamine. So if there's anything you could, again, association, substantia nigra, that's the dopaminergic center. Tegmentum is a juncture for dopaminergic pathway between the cerebrum to the cerebellum. So that's a passageway for the flooding of the signals. This is a pretty cool picture. Cross-section of the midbrain. Take the midbrain, you slice it open, and you look into it, right? And the one thing I think you can all notice is the substantia nigra spans almost the entirety of the border, right here on the left and right sides. Dark nucleus bundle. Right here, there's the red nucleus. This one, if you took anatomy 5, 6, 7 years ago, there's literally nothing about that structure in the book. They literally just give you red nucleus. More research is being uncovered. But the newer researchers point out that there is the activation. It coincides with another system called the RAS. And that RAS system is what kind of turns on consciousness. So when you're awake, the adrenaline synergizes with dopamine to give you alertness. It also plays a role for arousal, so behavioral, for sexual arousal, for being anticipating stimuli. Okay, so these regions are very closely linked. Then we have the pons. The pons is this bulging region of the brainstem. Highlighting a few of the key points here is, in order for the pons to connect to your body's motor for breathing, we have the middle cerebellar peduncle. That's the region that connects from the pons to the cerebellum. And then, of course, we have all the different respiratory centers. So another association I want you to have in your mind is pons, think of breathing, okay? More specifically, if you get into the weeds, the respiratory centers are called pneumotaxics and apopneutics. Those are two subdivisions of changing the diaphragm's contraction to change your breath. So if you're climbing a mountain and acclimating to a higher altitude, the diaphragm has to adjust. That adjustment is automatic. You don't have to tell your brain to change the rhythm. It just happens. But the way it happens is by this set of neurons detecting the change of CO2 and hydrogen and eventually changing the signal. When that action potential changes, then you change the rate of breath. Okay, so just remember, pons is to respiratory, of breathing. Okay, all right. Over here, have a picture. Bless you. So you can see right here, this is a big bulge. It's not actually that visible, but this region right here is what contains the apopneutic and pneumotaxic center in the middle. So right, it kind of spans from the pons to the medulla oblongata, and then right below that we have the spinal cord connected. Okay. One of the last items here is going to be your medulla oblongata, aka medulla, the most inferior portion of the brain stem. A few things I want to highlight. So again, whenever you're looking at these slides, just associate what structure has what characteristics. Number one, we have these pyramidal tracks. Pyramidal tracts are just the tract of the spinal cord as they come up from the body. They're going to route from that region to a dedicated part of the brain. Most of the axons are going to then decuss right here. So if there was any lesion or any damage that occurred in the medulla oblongata, then your body and your brain is disconnected. Okay. Likewise, there's a kind of a newer study on sleep physiology, which is really fascinating. When you're sleeping and you're dreaming, there's a point where the nerves are actually almost disconnected at a point. So you may be thinking something that's very vigorous or violent, and maybe your body isn't responding. And likewise, you might also have a violent dream. Some people sleep and they have this nightmare and they start throwing like a fit, or maybe you're sleepwalking, or you physically engage in the dream. This region contains two nuclei of importance. One is called the inferior. That means there's a superior one. But these nuclei relay proprioceptive information. Remember this word? What was that again? Proprioceptive? Exactly. Right. Time and space, it's going to be the distance. But basically, this is exactly what I said. When you're dreaming, and let's say you engage in a REM sleep cycle and you have this dream going on, it's violent. Sometimes people will act it out in their bed, right, in their sleep. They're actually moving. Some people just, when they're sleeping, it feels like your brain and your body are disconnected. So inferior nuclei, this is used for proprioception information. This is also what houses the inferior cerebellar peduncles. This connects the medulla to the cerebellum. And the last item for the medulla oblongata is for all of the somatic perception. Light touch, pressure, your fingers, tickling, all those stimuli for the sensory stimuli coming from your body or skin goes through your NCNG, the nucleus cuneatus and gracilis. They're all housed in the medulla oblongata. Okay, so association, as you're studying, medulla oblongata is where you decuss, crossing over of all the axons. It contains the inferior olivary nucleus because it's so on the bottom, it's on the inferior side. It also contains the inferior cerebellar peduncles because it's also on the bottom side. And the only one that's kind of the odd one is it's so low, so closely connected to your spinal cord that it perceives all of your light touch, your pressure, your skin. So you have your nucleus cuneatus and nucleus gracilis. Okay? As if you're just reading this, it's like a list of structures. Associate with key structures to the main item of the brain. All right, so I hope that makes it a little easier. One of the items that we quickly glance over is all of the autonomic functions, right? So I did just say that all the ANS systems, all the cardiac, the pulse rate, blood pressure, they're all regulated in this brainstem region. We're just going to call them centers. So cardiac center means anything that regulates your heart rate and also the force or the inotropic contraction. Vasomotor centers just means anything that regulates the pressure resistance. And then the respiratory centers just regulate the rhythm, okay? There is overlap with this part and the pons. So breathing is it's critical, but it's also it's not, you don't have to change the thought of breathing for your body to respond. So the pons and the medulla oblongata, they both are able to interact to fine-tune breath. A lot of it has to do with just the CO2 level, the proton levels. Okay? So cardiac center, vasomotor center, and your respiratory center. Nice and straightforward, nice and easy. Over here, this is a nice picture. Let's look at the one on the right first. This is a little easier. This is external view. Notice in this one, this is kind of a 3D imposed image. You can see some of the outlying traces of the nuclei. You have these big oval round, they look kind of like grapes, long, longitudinal. So these big oval structures you see, these are all the different sub-nuclei housed inside the pons. Within this region, you'll see that they have the cardiac center, vasomotor centers, you have the inferior olivary nucleus, but these different subgroups can change the rhythm. So for example, when you look at the ventral versus dorsal, one of them controls your external costal muscles, one of them controls your internal. And so that change of breath is fine-tuned, right? Because you hyperventilate, but eventually when you're up in the mountains for a week or 2 weeks, your body acclimates to it. The rhythm will change and you can feel a lot more normal. But the first time you ascend up to a certain altitude, you feel like you're gasping for air. You feel like you're choking because the diaphragm and then the signals have to match up. The rhythm has to sync up. On the left-hand side, notice the cranial nerve number 10. This is where all of the vagus nerve roots come from. And then if you look into it, you'll see the crossover. So this set of nerves cross over. This set of nerves cross over. Decussation. Right? So this is that key term, decussation. Okay. Don't look at everything because on this image you'll see some of the cranial nerves. You can just kind of cross those out, but just focus on what it's trying to tell you. Right. So this is where the crossing over occurs, and then on the right you can see the traces of the olivary and the different nuclei cells. All right. Cool. Last item. Okay. One of the final items on the brain is your cerebellum. This is the inferior 1/3 of the brain. It's a pretty massive structure by itself. The cerebellum is considered the second largest isolated piece of the brain. It is divided into several regions. There's 3 dedicated areas, and there are some anatomy associated with it, and you probably see a little bit of that in the HoloLens, but the outer layer of this region is called the cortex, just like most structures in the body. Adrenal gland, outer side is called the cortex. Same thing for the kidneys, outside it's called cortex. But the outer region is called the cortex. Within the cerebellum, there's this kind of lighter tree-like pattern of neurons called the arbor vitae. And this arbor vitae is the tree of life. This is a pattern of neurons. If you look, tilt your head, it looks like a Christmas tree. Within the deepest sections of it, we're going to have the nuclei. Okay, the cerebellar nuclei. Cerebellar just means cerebellum. And then it's partitioned in the midline to 2 hemispheres by a structure called the vermis. This is a dividing border. And these ridges and folds that overlap around the cerebellum are called the folia. Right, so again, a lot of terminology. That association, cerebellum, think about the different lobes. These lobes are called the folia. The dividing border is called the vermis. And the key function is this is for motor and gait. Right, so when you think about alcohol impairing the brain, this is usually the first site of alcohol damaging. And you also have Purkinje fibers that are the longest neurons, but the point is all motor functions. So right here, an image. On the right, you can see the size of the entirety of the cerebellum. The vermis divides the left and right border, the left and right hemispheres. And then you have the interior, posterior. On the left, you have a cross-section of the brain, the sagittal view. You cut it right in half. Tilt your head to the right. You see it looks kind of like a Christmas tree. Right? We just had the holidays, right here, kind of like a Christmas tree. And then you can see all the fibers. You can't see how long the neurons are, but isolated out, they have the single longest neurons in the brain. Okay, so by itself, one single strand, these are where majority of the longest neurons are housed. Okay, summarizing some of the key functions here is that it maintains fine-tuning of motor control. It also helps you process the memory of movement. So when you're dancing, learning an exercise for the first time, doing yoga, adjusting the muscle fine-tune motion, maintaining equilibrium, posture, and gait, and of course, proprioception. So this is the part that's most vulnerable to alcohol damage whenever you intoxicate yourself. In order for the cerebellum to talk to the brain, there are 3 connections I want to highlight. And these ones, pretty straightforward. The word cerebellar is in there. So if you miss this, if you can't associate cerebellar with cerebellum, that's not good. Okay, so there's a superior, there's a middle, inferior. All you need to be mindful of is a superior is on top, right? So that's going to connect your midbrain to your cerebellum, right? Because it's the highest one. And then the mid one is the pons to the cerebellum. And the inferior is the medulla oblongata. You need to always remember cerebellum is already on the inferior aspect. It's already on the bottom. So the superior part of the inferior part of the brain would be the midbrain, right? Just always know what position of the structure we're talking about. And then when we describe it with superior, middle, and inferior, you know what level of the brain we're referring to. Okay? So that way you're not just lost. All right. And then let me show you this one last thing here. This is just review. So remember, I think this is also towards the end of the nervous system we went over last year that I have the same slide. This is literally like 80% same thing, but the limbic system, right? The memory. Listed this out. And I know I had a couple questions on this already, but I probably won't emphasize this one on the exam because we have this already. But don't forget, one of the key items of the brain is your memory. long-term potential, the ability to form short-term versus long-term memory. That all happens inside your limbic system. A couple of structures that allow the brain to communicate include your cingulate gyrus. These are the ridges of the corpus callosum that allow you to bring emotion with consciousness. The parahippocampal gyrus is another region for association. So remember the word association means integration of sensory with motor to storage. Hippocampus is the structure that allows you to form the memory. So consolidation of long-term memory, LTP, that's long-term potential. These neurons utilize ions of calcium, magnesium to create a conduction that can then engage with another synapse, another neuron, and then overlaps with the amygdala body. Right? So emotion of fear. So when you're scared of the exam, that might trigger a stronger memory, right? Versus if you just, it's going easy, I don't care about it, it's not on your mind. And then don't forget that the olfactory bulb is directly connected, so you get to skip the thalamus tract to be able to trigger sense of smell. Okay, that's all I have for the brain. I'm gonna do some questions with you guys. Any questions on what I just went over for the brain aspect? Anything on the brain? Anything at all? So some of this might be a little bit of a recap. Some of it might be a little bit new, but I highlighted some of the key anatomical structures that I want you to know. There was maybe like 4 or 5 cadaver images in this set that will be on the practical, right? And then I'll give you guys a list of the 12 living structures I want you to know.